Okay. So welcome you all. Myself is Professor Dr. Deshpande, and I have muted your mics right now because I want to start the topic directly. And I request whenever I open your mics, please take care. If there is sound from the back side at your home or at your clinic, please close your mics. That is a basic requisite for using this software. Okay. So today is a Friday, that is the 3rd of April 2020, and we are conducting these online lectures for the PG students of CARC because there is unfortunately whole world is suffering from the corona pandemic, and that's why we have been locked down all the India by our Prime Minister. But to save our academic interest, I am taking these particular classes. As you know very well that today's, that means on the 3rd of April, I am taking the topic muscle contraction. So in spite of discussing any other things, I will directly share my screen with you so that you can understand the topic very easily and very nicely. Okay. So this is very interesting. That means the picture is very clear to you. If it is very clear to you, I can use my cursor and I can mark the points which are very important. So this is a very nice technique to teach any students. Okay. So this is a topic muscle contraction presented by Professor Dr. Deshpande, that is myself. And as you know very well, I think there are still some mics. Please close your mics. Prajakta Taru, please don't open your mic. Okay, that disturbs whole the scenario. So I may take this PPT. I'm just trying to take it in a PowerPoint presentation. Okay, so this will be more better. The big letters are there. So muscle contraction is the topic, and you can note my mail ID, as everybody has this mail ID, professordeshpande@gmail.com. This muscle contraction topic is from your syllabus. It is in the syllabus of UG. Here you can see UG paper 2. Because research is your, I'm sorry, this is UG undergraduate student, BAMS students. They have the 2 papers, and in the paper 2, in the part B, there is a point number 19, Physiology of Muscle Contraction. This was the title. This is the title in the UG syllabus. Now you are all PG. You have also the paper, Kriya sharir paper, as per your subject, because research methodology is the one paper and another is your own subject. So there is a prelim examination of MUHS. In that paper, you have the part A and part B. Part A is Ayurvedic and part B is the allopathic part. Of course, PG final students also get benefited by, because they have the paper 3, and there is also mention of the muscle contraction topic. Now, let us start how our muscles get contracted. This is contraction of my muscles of the palm, contraction, relaxation. This is simple to understand. But the process starts from your brain. Remember. This is the motor activity. Some points you must mention in your notebook, because I cannot write each and everything on this PPT. That's why I'm telling you, this is the part regarding the motor activity of your brain. Not sensory activity. This is motor activity. Muscles are concerned with the motor activity. This muscle contraction understanding is also a type of cellular physiology. I think I have used this word in the RMP and AP. I have told you that resting membrane potential, action potential, these parts are understanding at a cellular level. We are now understanding muscle tissue, but in that tissue also, we are going to see the minutest part of that muscle. Minutest part of that muscle, this is muscle fiber. Okay. So when the muscle contract, the stimulus is coming from the motor neuron. There are 2 neurons, sensory and motor. This is motor neuron. Jnanendriya, Karmendriya. This is Karmendriya. Remember, Pani and Pad in Ayurveda. Pani and Pad. We are talking about Maus Dhatu, Akunchana Prasarana. PG students are expected to know the allopathic part as well as Ayurvedic part and the relation in between Ayurvedic and modern parts. So don't forget to understand Ayurvedic and allopathic both the concepts. Maus Dhatu, Akunchana Prasarana. This is motor activity Karmendriya. Karmendriya related with the Pani and Pad, hands and legs, lower, upper and lower extremities. Now, the sarcolemma adjacent to the axon, this is the motor fiber axon, dendrites, sensory, axon, motor, dendrites, sensory, axon, motor. So this part is called as a motor end plate. Motor end, because that is the end part of that axon. And it is also called as a neuromuscular junction. Neuromuscular junction. Junction is a meeting place of what? This axon at the end, motor end plate is there. Now, these axon terminals at the end part, there are some bags, BAGs, bag, bulb. They are called as a bulb, electrical bulb. And that bulb structures are called as synaptic end bulbs. Can you see over here? I'm sorry. I will just maybe taken this one so that to mark it is very easy. So this is, you can see this is word, synaptic end bulbs. This marking is very important. And that bulbs, now this is a bulb. They contain some chemicals. They contain some chemicals and that are called as a neurotransmitters. This word is very important for our muscle contraction. This is the chemical molecules. And the important neurotransmitter for muscle contraction is ACH. Don't forget it. Muscle contraction, acetylcholine. Muscle contraction, acetylcholine. This neurotransmitter is very, very important. Okay, we will go further. Here I can move further. Okay. This is the structure of the skeletal muscle. This is one fiber, maybe like deltoid. This is a deltoid. This is muscle. What is there inside the muscle? There are the muscle fiber. Can you see over here? Muscle fiber. This is rod, muscle fiber. In the muscle fiber is a rod. And in that rod, there are microfilaments. In that rod, there are microfilaments. They are called as a muscle filaments. They are called as a muscle filaments. Okay. All definition. This group of the muscle fibers, they are called as a fasciculi. Fasciculi. This is cellular physiology. Understanding the muscle cell. I'm talking about muscle cell now. Remember. Okay. So skeletal muscle is formed by muscle fibers. That muscle fibers, ते लाकडाची मोळी बांधतो मराठीमध्ये आपण म्हणतो इट्स अ बंडल ऑफ वुड्स. Similarly, bundle is called as a, here you can see, fasciculi. And each of these fiber is surrounded by some membrane which is called as a sarcolemma. Sarcolemma is a membrane, don't forget it. It is a cell, always it is a cell. There is covering. Each cell has a covering, that is sarcolemma in the muscle. And cytoplasm in that is called as a sarcoplasm. So there is a covering, there is some material like sarcoplasm inside. Next. Now each these microfibril thread again has very myofilaments, very thin structures in it. Very thin structure inside, that is called as a myo. Myo means muscle. Filaments. Filaments is a very, very thin structure. Okay. So these filaments more over time overlap each other. There are two types of the filaments. I am going to discuss actin and myosin. You have heard these words. They always overlap with each other. Sometimes there is a great overlapping, sometimes there is a faint overlapping, no overlapping. So there are, when there is an overlap, this is a dark band, dark. No overlapping. That is called as a light band. Can you see over here? Alternate. Dark, light, dark, light, dark, light. So there are inside the muscle. We are talking inside. There are dark and light bands. Okay. Next. Here. Now these bands are described in this way. The dark is A band. It is called as A band. Dark is always A and light is I. Dark is A, light is I. Don't forget these words. In the oral, examiner will ask you, what is this A band? So A is equal to anisotropin. Anisotropin. Can you see anisotropin? Huh? No light is going because this is a thick. So light cannot pass through like this. When it is a thick, light cannot go. That's why the word is anisotropin. And when light can pass, can you see here? Isotropin. Only thin. So light can go. Light can go. Dark, light don't go. Light can go inside. This is the simple thing. Dark band is equal to A, light band is equal to I. Now, these myofilaments are attached to a narrow zone in between that sarcomere, the muscle structure. There is a one Z line. This is imaginary line. Actual no line is existing over there. Scientist has given the name where the lines are. So this line you consider as a Z line, like PQRST waves. So it's just a naming given by the scientist. Actually, as such, no line is existing. It is a marking where they are meeting each other. Okay, and H zone lies in the center of A band. Now, this understanding is only with the figure. Now, I am going to show you figure afterwards. That figure you must remember in your mind. Otherwise, this description you cannot write in the examination. While looking to the figure, you can write down. Okay. So halfway along the length of I band, myofilaments are attached to a narrow zone that is Z line. And two words you should remember. Number one, Z. Number two, H zone. Write down these words, Z and H. I will show you where it is. Can you see? These are the Z lines. These are the Z lines. And they are coming here in between. This is only the thick H band. Okay. And this is A band. Can you hear this? Again, A band. H zone is consist of the A band only. You have to remember this figure by heart. There is no choice for it. Otherwise, you will not understand. Only you have to remember this thick filament is called as a myosin and this narrow filament is called as a actin. There are two words, myosin and actin. Myo, myo. Look my action. Myo. O is there. O. Myo. So myo is a thick. Actin. Thin. Actin. Actin. How to remember? Myo. Thick. Actin. Thin. That's why you can remember only with my action. Remember in college, remember my action. Myo. Myosin is a thick. Now, these are the contractile proteins. These are the contractile proteins. That is these are proteins. Remember. That's why we say shiryate tachariram. Whenever there is a degeneration, whenever you want to build up the strength of the muscle strength is very important in our practice. Hemiplegia, loss of strength. Why? Here you can see. Proteins. These are proteins. So you must eat the green gram, turichi dal kha, soybean kha. You must eat the soybean, green gram, black gram, urid, mashatayna samwahana masas, mashatayna abhyanga. Why? This is proteins, myosin and actin. Okay, and then 200 molecules of myosin together becomes the thick filament. There are total 200 together and that forms the thick. I think you have seen this figure, thick. This is made up of the 200 molecules. Okay, and this particular myosin structure, you must remember this is simile. They are looking like golf stick. Golf stick. Have you seen golf? You know the game golf. So that is like a hockey stick or golf stick. Hockey stick or golf stick structure. Okay. Then next. So thin filaments are called as actin. But actin is more complicated protein. Actin is more complicated protein. Remember, this is not written over here. Complicated. Why? Because it has again additional proteins. Actin contains, actin filament contains. Actin is itself is a protein, made up of protein, but it also contains, write down these names, troponin and tropomyosin. Troponin, tropomyosin. Basic only two filaments. Remember my basic only two. Myosin, actin. But in actin, there are again troponin, tropomyosin. There is addition. Don't forget this. Huh? This tropomyosin, this particular protein, covers the active site of G-actin. This is a covering. Tropomyosin protein covers some part of the actin molecules. Tropomyosin covers some part of actin. So always it is closed. Actin is always closed. I will not allow myosin. That I am closed. But with some energy, it is closed. It is opened. It is opened. Actin opens. I will show you how it is opened. Okay. Now, I think that we have seen troponin in the protein which pulls tropomyosin to cover and uncover. Troponin, what the function of troponin? First, I mentioned tropomyosin is a cover. Where? On the actin molecule. Okay? Tropomyosin is a cover on actin molecule. Now, this troponin, what is the function of troponin? It helps to cover, uncover, cover, uncover, cover, uncover. Who is doing? Can you see here? Troponin. So, troponin is helping to tropomyosin to cover and uncover actin. Basically, only two. Huh? Don't write in the exam 4 or 5 proteins. Don't write. Only 2 filaments. Now, this troponin has 3 parts. Little bit complicated, I know. One after another, one after another. Troponin has 3 parts. I, insignificant site. T, site for tropomyosin to attach. And C is a site for calcium entry. ITC. Mug it up. ITC. This is a part of troponin. ITC. R is a no value, insignificant. T is a site for attachment. And C is the site for calcium entry. Dr. Sagar, are you tired? Because it is looking to me directly, I can see you, okay? Because that will also be seen in the YouTube. So the people will say, oh, Dr. Deshpande's lecture is going bored. Because Sagar is giving, oh, no. So my request, if you are tired, you can go out and again join, no problem. Okay, this is myosin. This is myosin. Golf-like, hockey stick. Basically, here you can see, this is a hockey stick. This is a handle and this is a part. Hockey stick. Can you see hockey stick? Hockey stick. Here, myosin. They are twisted like DNA structure. They are twisted to each other. Myosin filaments. Okay. How you can? These all figures you must draw in examination. These all figures you must draw in the examination. Remember my words. This is the actin molecule. And here it is a troponin and tropomyosin. This is all. As I already mentioned, there is additional of the proteins. C. Can you see this troponin has a, I have already mentioned, I is a insignificant part. C is for the entry of calcium molecule. Calcium is very essential. Acetylcholine and calcium are very much essential for muscle contraction. Don't forget, neurotransmitter ACH plus calcium is very, very essential for muscle contraction. Can you see this is all is a actin molecule? Can you see actin? Chain of actin. And here this is myosin is coming. This is the head of myosin. Write down this word, head of the myosin. Head of the myosin is very important. Hockey stick. Hockey stick. Head of the myosin. This is attaching to the actin. This head is attaching to the, see my cursor. Head is attaching to the actin. Head is attaching to actin. Okay? Let us go further. See, this head of the hockey stick, head of the hockey stick, this is configuration. What is meant by configuration? Actin and myosin together. Actin and myosin for muscle contraction. So this is the head, don't forget it. And now, actin molecule, before it was closed, now it is uncovered. Now it is uncovered. Whenever there is uncovering, then only this can attach. Is it clear? Only actin molecule when it is open, my doors are open, come myosin, come to me, attach me. Then only it will come. If actin is closed, myosin will not attach. Is it clear? Actin is opening the door. Come on, take me out. So myosin is a very strong, is a thick. This is very thin. Next, from the cerebrum, actual. This is. Can you read? Actual mechanism. What is happening? 1, 2, 3, 4, 10 steps. One, current is coming. Neurotransmitter is there. ACH there. And what is? Do you remember, Sagar? Depolarization takes place. RMP. Whenever there is a depolarization, can you see here? Action potential. Cell membrane polarity change. And now let us start. Action has been started. Next, impulse is conducted to the T tubules. This is the sarcomere structure. In the muscle, there are some srotasa in Ayurveda. Srotasa in mouse dhatu. That is T tubules. And also it spreads all over the reticulum. All covering is there. The current is spreading all over the current. And then what is happening because of the current? Calcium ions enter into cytoplasm. Very simple. From the membrane, calcium ions enter into the inside fluid, cytoplasm. And so what is happened after that initiation? There is after attachment of calcium, remember this thing, only after the attachment of calcium, troponin rotates and actin is open. Okay, binding between troponin and tropomyosin becomes loose, and tropomyosin is exposed. Open the door. Then next, active sites of G-actin becomes uncovered, and head of the myosin comes in contact with the active site of G-actin. Can you see again here? Here, this is actin molecule. This is the open site. This is uncovered, and this is covered. Can you again go back? Here I have shown you this picture. This is G part and this is the troponin part. So this figure you must remember. Okay. Next, what is happening? When myosin head bind to the actin, produces power stroke of contraction. It's like a stretching. With the head of the myosin, it is pulling the actin molecule. Myosin is strong. Myosin is pulling active molecule of actin. It is clear to you? That is called as a power stroke of contraction. Then myosin head moves toward the center of sarcomere, like the oar of the boat. Can you see oar? मराठीमध्ये त्याला आपण ते काय वल्लव म्हणतात ना, वल्लवण्यासाठी बोटीला ते आपण वल्लवतो मराठीमध्ये. That is English, it is called as the oars of the boat. While you drive the boat, it's like taking ahead and ahead and ahead. Again, I will go back and show you the figure where it is going. See, we have to take this molecule here. This molecule here. Are you understanding? This molecule should be brought to here in the central line. Pulling this part to this part. Pulling this part to this part. And this part to this part. So what will happen if the two things are coming nearer? That is contraction. Is it understanding to you? Okay. Now, so The myosin head moves towards the center of the sarcomere like the oars. This action draws the thin filament past and thick filaments towards the H zone. I think I have shown you that H zone. Here we have to bring H. Can you read it here? H. We have to bring this to this and this to this. Who is bringing? Myosin is bringing to actin. Okay. Now, you remember the slide number? Okay. I think we have conducted. This is also complete. This is now. ATP liberates energy. Remember the who is doing the energy? ATP. ATP molecules are there in the muscles. They break the actin myosin complex and pushes the head of the myosin to the next side. That means, again I will go back here. This is the first part, okay? This is the first part. Then ATP, again energy coming, this coming to this part. Again energy, this part will come to here. Again energy, this will part come to here. So myosin from here to here, even this action. Can you see? Myosin will move here, 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 here like this with the energy of ATP. From this molecule, and here it is finished. Here it is finished. Contraction process is finished. When all last molecule is attached, finished. And then what next? It's a relaxation. Always there is a cycle in the muscle, contraction, relaxation, contraction, relaxation. Remember always. So this is the process of contraction, and after that it is relaxation. Again, we will go back. It continues till the last site is available. Remember this, till the last site. Can you see again over here? What is the last site? This is the last site. This is the first site. This is the last site. I think I am trying to clear as much as possible. This is the complicated actually topic, but I am making it very easy for you. Okay. Immediately after depolarization, whenever the contraction process completes, then there is another enzyme, cholinesterase. ASE means enzyme. This is another enzyme. What is the function? That is in the synaptic cleft, where it is in the synapses. What it does? It is destroying the acetylcholine. Cholinesterase stops. ACH, no, it is enough. Contraction is complete. Contraction is complete. Who will act? After contraction, who will start functioning? Cholinesterase. What is the function? Cross on ACH. No ACH. When ACH stops, what will happen? Calcium ions are reabsorbed. Remember the term. Calcium ions are reabsorbed. Okay. And as there is no calcium, what will happen? Again, troponin and tropomyosin interaction stops. Troponin tropomyosin interaction stops. Again, it is closed. Active molecule is closed. Everything is repolarized. They say as it is before. So this is the cycle of contraction. At the end, what will be there? Relaxation. Okay. So these are the different contractions which we see in our practice. Some are a twitch. They are called as a twitch. Titanus, dhanurvata. There is also severe contraction. You must tell me tomorrow what is meant by isotonic muscle and isometric muscle contraction. There are two types. While you advise to the patient exercise prescription, you must know walking. What is that? Is isotonic or isometric? Weightlifting. What is that? Isotonic or isometric? I will give you the hint. Tone means, iso means same. When after contraction, muscle tones remain the same, that is isotonic. If the metric means length, if the length of the muscle fibers remains same, iso means same, that is called as isometric. These muscle contractions can be recorded on myogram. Next, muscle tone. This concept is very important because this is a sustained partial contraction. Now I have sit in my chair. I can sit. Professor Deshpande can sit in his chair because of my back muscles. My back muscles, very good muscle tone. Very good muscle tone. If my back muscles are not strong, I will fall down. I will fall down. So muscle tone should be good when you want to maintain Can you see here? Posture. Standing posture, sitting posture, walking posture. Who is maintaining? Muscle tone is maintaining. And what is the opposite word to the muscle tone? Shaithilya. Mausa dhatu. Shaithilya. In Ayurveda, it is called as the Shaithilya, lack of tone, that is flaccidity. Okay? Next. In the muscles, we get the different types of the disorders or the problems in our practice, and that can affect the muscle contraction. That's why we have mentioned it. That can affect the muscle contraction. One is atrophy, that is wasting of the muscle. With the disease, with the improper diet, then there is a hypertrophy of the muscle, increase in the diameter. These two words are opposite, atrophy and hypertrophy. Then fibromyalgia. Fibromyalgia, this is non-articular rheumatic disorder. There is no connection with the bone joint, no connection with the bony joint, only myalgia. Myalgia means pain. Gia means pain. Pain related with the muscle is called as a myalgia. And when there is a fibrous tissue formation, fibromyalgia. Okay, what are the complaints? Pain, tenderness, stiffness. Pain, tenderness, stiffness of what? Muscle, tendon, and ligaments all are involved. Muscles, tendons, and ligaments. Common example is backache at the lower region, that is lumbago. Okay, and last is very important disease, that is Myasthenia Gravis, by which our great Bollywood star was suffered few years back, that is Amitabh Bachchan. He has suffered from Myasthenia Gravis, MG. That is great muscular weakness and fatigability from the improper neuromuscular transmission or deficiency of, I'm sorry here, printing mistake, ACH, acetylcholine. Deficiency of acetylcholine is equal to Myasthenia Gravis. Very important. First symptom of this disease, anybody knows? It's a drooping of eyelid. Drooping. Can you see on the screen? Drooping. Left. Drooping. Close. Drooping of the eyelid. Drooping. This is the first sign of Myasthenia Gravis. Okay. Then what are the abnormal contractions? What are the names for the abnormal pathology? That is spasm, abdominal spasm, spasm in the hand, spasm in the leg, spasm in the calf muscle, tremors, fasciculations, fibrillations, and tics. These names you should remember as an abnormal contraction. So my dear friends, I have tried my level best to give you the very good idea about the muscle contraction topic, which is very important for the medical practice as well for your examination as a long answer question. I think you must tell me, I think today's video is a little bit more clearer. My both the sides of the face are a little bit bright. Otherwise, I always a one-sided person. Okay, I stop sharing and then I can unmute all. So there are today 6 participants were there. In general, everybody should answer me very fastly. Prajakta, have you understood the concept?
Yes, sir.
Dr. Sujitra?
Yes, sir.
Dr. Yuvraj?
सर ते पीक म्हणजे काय ते विचारायचं होतं.
No, at the moment I am asking you, whatever I have told you, is it clear to you or not? We are not discussing here the question. We are not, because this is a limited time span. For my software, I am not going to answer.
Lecture is really good. Excellent.
Thank you very much. Dr. Sagar, he is maybe busy at the moment.
Yes, sir.
Okay, and I think there is Shantanu also. Mr. Dr. Shantanu, have you got the clear understanding of muscle contraction? So you will be able to write the question in your exam? Okay. So I thank you very much, all of you, and we will meet. I will request all of you, you can please write down all your questions, and together, when after a week, you can write me down in the WhatsApp. I will collect it, and only one lecture will be there regarding only question and answer, solving the difficulties. Because even I have corrected my formula, Previously, I used to ask Dr. Sagar, say something, Dr. Suchitra, but that is something I have found it is a wastage of time, because then I cannot manage the time to complete the topic. On Monday, tomorrow and Saturday and Sunday, there may not be lectures. You can have your own family problem things. And then from Monday, again, we will start at 12 o'clock. Monday, there will be research lecture. I will give the topic afterwards. So once again, thank you very much.
Okay, sir.
And enjoy your whatever the profession you have. Thank you.
Thank you.